So much of the extraordinary biodiversity that we see on this planet can possibly be attributed to the fact that organisms experience heterogeneity in their kinds of ecological interactions. And just to give some classical examples of that, on this side over here we have stickleback fish which have diverged in their body form in response to different feeding environments. This has been thought to have been spurred on by competition. And over here we have Tamima walking stick insects which have diverged in terms of their body coloring in response to living on two different host plants. And this obviously reduces predation on these animals. Now, pollination biologists too have been thinking about these ideas for a long time. People like Grant and Stebbins have envisaged how plants which experience different pollinators in different parts of their ranges may diverge in floral form. And so today I'm going to be talking about a wonderfully interconnected group of pollination systems. And I'm going to try and show you a very graphic example of how pollinators drive both the convergence and the divergence of floral traits. And the organism linking all these systems together is this long proboscis fly over here called Presention longitennis. Now, this thing here is actually a proboscis, and it's used solely for the purpose of extracting nectar from very deep tubes. And it can vary in length depending on population. It can go as low as 20 millimeters and as long as 70 millimeters. It pollinates quite a wide variety of flowers. We know of at least 18 species which appear to be partly dependent on this fly, and these are some of them featured here. You can see some similarities between these plant species, often from very different families. And you can see that they're often similar colors, sort of white to pink, and they mostly have long tubes. The tubes tend to match the lengths of the pollinator's tongue. So here's a graph just showing that. You've got pollinator proboscis length over here, so it's just this one species of pollinator. You've got average flower depth on this axis. Each one of these points is a different site. And at each site there may be multiple plant species involved. So actually these are just averages of all the different plant species that these flies pollinate. So it gives you the idea that perhaps at each site, the different plants at each site appear to maybe be converging on the tongues of these flies at each site. So there may be some convergence going on there. What I find perhaps even more interesting is that if you take these 18 species of plant, at least 10 of them occur outside of the range of this fly. And when they do occur outside of the range of the fly, they look different and they're pollinated by different pollinators. So these are 10 of the species that occur outside of the range of the fly. This is what they look like in the populations where the fly is present. And this is what they look like when the fly is not present. So, for example, just to illustrate a few of these, this is a bee-pollinated gladiolus marklei. This is gladiolus marklei when the flies are present. So differences in color, slight differences in tube length. Ethan Newman has spent a talk on marine humulus showing how long-tongued flies and short-tongued proboscis insects select on different style lengths. These two dices over here, the one on the left is pollinated by flies, the one on the right is pollinated by butterflies, and you can see differences in color as well as orientation. Then we have a whole lot of different pelargoniums over here, which differ in tube length depending on what pollinator visits them. And I'm not going to talk any more about those because it'll get a bit tedious. What I'd like to do now, though, is just say this possibly give the idea that shifts in pollinator are driving divergence in each of these species. But I'm not going to talk more about the entire guild. What I'm going to do next is ask you to forget about the rest of the guild and focus in just on one species of plant. So this plant over here, Tritoniopsis revoluta. And what I'm going to do is I'm going to hopefully convince you guys that this fly is playing a role in driving the convergence and divergence of floral traits, in particular tube length. So this plant has an interesting distribution range. You can see two little populations here in the north in that long string of mountains. You can see a bunch of populations in this mountain range, and you can see a very isolated population right down there in the south. You'll also notice that those study site symbols are different sizes. So the different size circles represent tube lengths of different lengths. Okay, and you can see a fair bit of variation, especially right there. And if I show you pictures of these flowers, they're radically different. Those populations are quite close together. This flower has a tube length of 70 millimeters, and this one of 10. If I show you what pollinators we've caught in these populations, all of these populations are visited by Amygdala bees, which have short tongues of about 10 millimeters. So here's an Amygdala bee visiting. Of these populations, we have found Amygdalas in 2 populations and nothing else. So in those 2 populations, there are only Amygdala bees as far as we know. In the rest of the populations, they're all pollinated, in addition to the Amygdala bees, by long proboscis flies, but by different species of long proboscis flies. So for example, in this population here with the very, very short tubes, there's a short-tongued long proboscis fly, with a tongue of about 10 millimeters that visits it. In all of those populations, there's a very impressive fly, this thing over here, Proseca longipennis, and in all of those populations, the fly's tongue is about 70 millimeters. In that population over here, there's a third species of long-tongued fly, and his tongue is about 30 millimeters long. And in that final population over there, there's a fourth species of long-tongued fly, and its tongue is about 20 millimeters long. So you can see all this variability in the pollinator landscape. And when you match the tongue lengths of the fly pollinators with the tube lengths of the plants, they marry up beautifully. I mean, I couldn't have hoped for a better now, these are just the fly pollinator populations. If I put in the 2 bee populations, they're not quite fitting in. So it doesn't all work out perfectly. You can see the bee tongue lengths tend to be quite a lot shorter than the floral tube lengths of those 2 populations that they visit. So what I'd like to do now is try to add the perspective of geography and relatedness to this whole picture. So this is a haplotype network just based on 2 fast-evolving chloroplast markers. When populations have the same color, it means they have the identical haplotypes. The numbers between them are just numbers of base pair substitutions. What you can see here is that in the north there seems to be one lineage, in the south there seems to be another lineage, and there's at least 2 very distinct lineages in those mountains over there. And again, the circles are just showing the different sizes of the tubes. So now what I'd like to do is try to overlay all of these 3 things. So we've got the phenotypic matching between plant and pollinator, and I want to overlay relatedness as well as geography. And I'm going to start with this regression again. At the moment, it's a bit unsatisfactory because you don't know which pollinators are in which population, right? So what I'm going to do is, instead of having dots here, I'm going to put the pollinators on this regression. So here you have them. Here you see one species of long-tongued fly, okay, visiting all those sites. Here you see the other 3 species of long-tongued fly. And I'm just showing the differences in the species of long-tongued fly by changing their orientation. I hope you can see that. And then over here, you've got those two little Amygdala bee-pollinated populations. So now you've got a little bit more information about what pollinators in each of these populations on this regression. Now what I'd like to do is overlay this idea of relatedness and geography to see how this affects the whole pattern. And to start with, I'm going to show you some of the less interesting haplotypes. So over here I've overlaid this purple haplotype. So it's these northern populations. Now I'm going to overlay this southern haplotype. So that's the red population over there. Where it gets really interesting is where I start overlaying the populations in the middle of the range here. So I've now overlaid this green haplotype. You can see three populations over here of the green haplotype, all pollinated by the same pollinator, all having very similar morphologies, really, really long tubes. But then you have that population there, extremely closely related, but very, very different in terms of morphology and pollinated by a different pollinator. So it suggests that these very closely related populations have diverged in response to being pollinated by very morphologically different pollinators. Now I'm going to overlay this blue haplotype over here. So there you have the blue haplotype, and again it's showing something interesting. Here you have two very, very distantly related lineages looking morphologically very, very similar and pollinated by the same pollinator. So it's suggesting that there's ecological convergence here. Two distantly related lineages have converged in tube length in response to being pollinated by the same pollinator. The last thing I'm going to do is introduce this little population over here. It's this tiny little blue guy over there. It's very, very closely related to these blue ones, just one base pair substitution difference. And again, you've got this incredible difference in morphology in these very, very closely related populations. And again, in response to a shift in pollinator between a short-tongued pollinator and a long-tongued pollinator. So here you have ecological divergence, where closely related lineages have again diverged in response to having different pollinators. So in conclusion, distantly related lineages appear to have converged in response to having the same pollinator. Closely related lineages often appear to have diverged in response to having different pollinators. And with that, I'd like to thank the people who've been involved in this project: Ethan Newman, Petra Ross, and Alan Ellis. In terms of phenotypic traits, so
other phenotypic traits other than chlorophyll.
Other than tubular.
Yeah.
So flower size tends to differ, and it seems to correlate with tube length. No, the vegetative traits are really simple. Can't really see much there.
Similar habitat?
Tend to be arid habitats, but yeah, similar.
What prevent migration? Like seed dispersal?
Seed dispersal is very low. There don't seem to be any particular adaptations to long distance dispersal, so the seeds fall very close. They're quite discrete, the populations. Really, I think it's only through pollen flow that you're going to get genes around.
How quickly do you think these populations will divert?
I'm not 100% sure. Yeah.
Fast or slow?
I would expect quite fast. Yeah.
So it looks like the plants chase the flies, at least on an individual species basis, that maybe the flies are adjusting to the community of plants that are there. Do you have any
I think
evidence for that?
Is that
That's
okay.
fine. Yeah.